Good morning, members of the Sweden community. I apologise for my absence. Nevertheless, I will still present my findings and pitch my need for further research funding of the genetic disorder known as cystic fibrosis, or CF. This particular inherited genetic disease is caused and developed due to a direct result of changes upon the DNA of human chromosomes. It can be identified early from birth and can be spotted through 5 main signs, including that of salty sweat, thick mucus excreted by the body, sinus difficulties, abnormal pancreas functioning, and trouble breathing. The disorder carries along with it symptoms that persist inevitably. Majorly, sufferers experience a cough, of which is quite intense, normally involving blood and phlegm, respiratory issues like shortness of breath, wheezing, and pulmonary hypertension, due to blocked airways, as well as gastrointestinal problems concerning excessive heartburn, diarrhea, and constipation. Some other minor symptoms include whole body fatigue, abdominal pain, delayed growth, and infection of lungs. The genetic change by which cystic fibrosis occurs is present within the human chromosome 7, inside the CFTR gene referred to as a cystic fibrosis transmembrane conductance regulator gene. This particular gene houses over 200,000 deoxyribonucleic acid nucleotides, of which are responsible for producing the CTFR protein, allowing the human body to flow chloride ions and water through the cells. This ion channeling is very significant as an incompatible amount of ions flowing causes epithelial surfaces to contain unsafe amounts of salt and water. This is the basis for cystic fibrosis, whereby a genetic mutation in the development of the CTFR gene impacts the CTFR's protein structure, leading to unhealthy amounts of salt and water passing into the lungs and pancreas, producing unhealthy amounts of mucus, responsible for blocking of airways and reduced functioning of the pancreas. The genetic disorder itself is recessive, by which it is carried as a recessive gene through individuals and continues to be passed down through generations. Our research shows approximately 1 in 25 individuals are carriers of the disease, meaning out of the 2 CTFR genes, one is mutated and the other is normal, by which the normal gene compensates for the other, blocking cystic fibrosis from being shown. However, the gene is still present, and those individuals who inherit 2 mutated CTFR genes are those born with the disorder. This Punnett square our team has devised shows exactly this information, by which a mother and father both possessing this recessive trait of the disorder show the chances of their offspring having the disease. The information shows that there is a 75% chance that the child will be unaffected but will carry the mutated gene, and that 25% chance that they will have a child with both homozygous recessive chromosomes, meaning that 1 in 4 possibility will be born with cystic fibrosis. Also, a family pedigree my scientists have created portrays this information efficiently. As you can see, the disorder is carried recessively, with the first generation being carriers, with 1 of 4 children possessing homozygous chromosomes. Also, the third generation individual has a homozygous pair, of which skipped a generation as those parents were also carriers, and passed down both recessive traits to their daughter, causing her to have cystic fibrosis. Diagnosis for CF is not severely difficult or painful, as it mostly involves only a couple of tests. There are 2 major examinations that can be undertaken, of which one is carried out at birth or soon thereafter. A newborn can be genetically tested through a simple blood test whether they contain any faulty CTFR genes, and if so, whether it is the homozygous pairing or recessive heterozygous chromosomal pairs. The common and most effective diagnosis procedure for CF, however, is the sweat test, involving a doctor collecting samples of sweat excreted from a person's body in order to test the salt levels within the sweat. If the results prove there is an abnormal amount of salt, then the individual is diagnosed with the disorder. Unfortunately, patients suffering from cystic fibrosis currently have no cure, but there are numerous treatment options to aid in reducing certain symptoms. The 3 major preferences specialists suggest to sufferers include medicine, through prescribed antibiotics and anti-inflammatory drugs to reduce swelling of airways and to treat lung infections. Chest physical therapy is highly recommended but is difficult for some users. It involves slapping the chest multiple times whilst lying down, in order to loosen mucus inside the lungs and airways that is then coughed out. Lastly, CF sufferers may need to resort to oxygen therapy if breathing has become too difficult, of which oxygen is injected directly into the lungs through a mask and oxygen tank. However, sufferers are limited, as medicine will only reduce the excess mucus and will never fully replace it back to normal, causing lifetime problems with lung infections and breathing. Also, the many medicines required are very expensive. Mucolydotas and mucolytics required in treating the disease are in excess of $70,000 for patients. Currently, there is one effective method using biotechnology aiming to completely rid CF from sufferers called gene therapy. It is a difficult process and involves cloning correct CTFR genes and replacing mutated genes in the DNA of cells and airways with them. So far, scientists have been unable to successfully allow the inserted genes to produce chloride ions to the extent of a normal person, but excess salt production is severely reduced, seen in the trials conducted. In summary, cystic fibrosis is crippling to sufferers, and with the treatment options present today, there needs to be drastic change if we want to rid CF completely. This is why I ask you to supply my team with this funding in order to research into the chloride ion channeling and finally crack how gene therapy biotechnology